Sir, recombinant DNA technology is just a minute, beta. In here, basically, we have studied few diseases till now, like we know diabetes mellitus, we know certain like bleeding disorders like hemophilia, we know sickle cell anemia, isn't it? So there are certain disorders which are genetic. That means by birth itself, like type 1 diabetes is inherited. That means right from the birth, the person may have such defect. So in such condition where like bleeding disorder like hemophilia, you see like the person will be continuing to bleed without any stop. Generally we see only in case of males. Color blindness, you see it is seen only in males. So there are certain genetic disorders because of that defective genes. What you will see, that particular protein will not be synthesized. For example, in case of hemophilia, we don't have the clotting factor 8 synthesized. We don't have clotting factor 9 synthesized. In case of diabetes mellitus type 1, we don't have insulin synthesized. So in all such genetic disorders where a particular hormone or a particular substance is defective, we can use certain techniques in lab where we are trying to synthesize these things naturally by making use of certain natural agents. So that's what is recombinant DNA technology. Okay, so that recombinant DNA, I'll just see definition, you will understand if you understand this. See here, the donor DNA is taken. The donor DNA can be a plant DNA, it can be animal DNA, it can be bacterial DNA or any donor. Because we know genetic code is coding for the same amino acid. It is universal. AUG will always code for example, alanine. Whether it is for plants or whether it is for animals or whether it is for bacteria or whatever, that particular codon, that particular code word is always constant for that particular amino acid. So you are selecting a donor DNA. For example, you are selecting a bacterial DNA or animal DNA. Okay. Now you are just trying to cut a part of it, fragment. For example, a part of the DNA which is coding for insulin hormone. You are just cutting that particular part of the DNA fragment from donor. And now what you will do, in order to cut this DNA fragment, you use the enzyme restriction endonuclease. A part of DNA fragment is cut from a donor. For example, animal DNA. Now what you do is you are just inserting into a biological machine. For example, this is bacterial DNA, which is plasmid. Extra chromosomal bacterial DNA is called as plasmid. Plasmid is bacterial DNA. Now you are just cutting, opening this plasmid with the help of same enzyme, restriction endonuclease. You just cut open it. Okay. Once you have cut open it, you are making use of enzyme ligase and you are just introducing this desired DNA piece over here. Okay. And now you are getting a new DNA which is called as recombinant DNA. Okay, now this bacterial plasmid will have a particular genetic information which is coding for example, insulin hormone. So what you will do, this recombinant DNA, now you will insert into a bacteria, any bacteria, okay, which will have enzyme machinery to synthesize that particular protein. Insulin is a hormone which is chemically protein in nature. Clotting factors, they are also protein in nature. Most of them are basically proteins in nature. So you are finding this recombinant DNA is now introduced into a host cell. The host cell can be animal cell, plant cell, bacterial cell, which will have enzyme machinery. Enzymes are available so that from this information, the required protein is synthesized. Now, once you are introducing this recombinant DNA into any living host cell, it will start synthesizing that particular protein. See, protein encoded by this cloned gene. Okay, so this mechanism is actually called as recombinant DNA technology. A piece of DNA is cut from donor, it is introduced into a plasmid. This plasmid is reinserted into any of the enzyme natural living machinery, whether it be a plant cell or animal cell or bacterial cell, which will synthesize the required proteins. So this is nothing but recombinant DNA technology. Okay, now once you write the details in your own words, once you understand this, what you do is, see here, now the most commonly used enzymes we can give, like enzymes also can be introduced into this genetic engineering. So few enzymes you can write: alkaline phosphatase, it removes phosphate groups. Two or three are enough, better. DNA ligase joins DNA molecules. Okay, DNA polymerase synthesizes DNA complementary to DNA templates. Like simple things you can choose. Now see here, some example of host cells which are used in recombinant DNA technology, that is genetic engineering. We can make use of bacterial cells like E. coli, Escherichia coli, Bacillus subtilis, Streptomyces species. We can make use. We can make use of eukaryotic cells also, beta. Like we can make use of fungi. Few examples like Saccharomyces, Aspergillus. We can make use of animals like insect cells, oocytes, mammalian cells. We can make use of plants. Whatever, all these can be used as a machinery in order to synthesize the required protein. So these are all host cells. Okay. So then, so all these I am scrolling so many pages. You should make a note because it's given as an essay, and this chapter itself is of 15 pages. Not required. Whatever I am telling you, you elaborate only that. Okay. So that those, for hosts we have bacteria, we have fungi, we have animal cell, we have human cell, whatnot, isn't it? Then see here, the different cloning vectors with the corresponding host and the size of foreign insert DNAs. No, suppose host is bacteria and you have used vector, that means cut plasma agent. It is Phage delta in case of E. coli. So E. coli will be able to synthesize a protein size like foreign inserts, DNA size. How much we can insert? 5 to 25 kilobase pairs, KB. Like that, see, east chromosome is the vector. Okay, so in the host is this S cerevisiae, and the size of DNA insert, how much? It's quite big, 200 to 2000 kilobase pairs of DNA fragment can be inserted in this particular host, and vector is this. Okay, then after this what you do is, there is one table which helps us to understand what are the uses. That table you choose, that's enough. See, insulin is synthesized by this genetic engineering technique. Okay, so the trade name, what we are synthesizing by this recombinant DNA technology is humulin. And why do we use this recombinant DNA product? To treat diabetes mellitus. Growth hormone, trade name what we are using is this protropin, humotrope. So we use this in case of pituitary dwarfism. Like that, we have few examples. And because he is asking uses, no, it's better you give at least 6 to 7 examples. Better. These are the names of diseases. These are the trade names of the rDNA protein which is synthesized. Okay, and this is the rDNA product, trade name, and what is the disease. Okay, so that is all about recombinant DNA technology. Actually, uric acid we have already studied, so we will replace some other essay for this. Uric acid we did, isn't it? Purine catabolism, uric acid, adenine. We have seen like it is broken down to xanthine, hypoxanthine. Finally, you will see uric acid. What is the drug of choice to treat this? Allopurinol. What is the name of the disease? Gout, isn't it? Accumulation of uric acid crystals in the joint spaces. Those crystals are called as tophaceous gout. So allopurinol is a drug of choice. Okay, we will do one answer from protein metabolism. Okay, so that we can replace this question. Let me see like which question is better. Why is it called sunshine vitamin? Because it is found abundantly in sun rays. That means with the help of sun rays, our skin will synthesize natural vitamin D. Okay, so vitamin D is fat soluble vitamin. It resembles steroids. Okay, so that means we can also, like vitamin A is considered as hormone. Even vitamin D is also considered as a hormone. Okay, so in the plants we have ergosterol. So in the presence of sunlight, you will see ergosterol is converted to ergocalciferol. Okay, so chemistry you need not know, but nobody will ask usually. Okay, so synthesis. See here, this is very good picture which but you have to understand. See, in the animals like in the skin we have 7 dehydrocholesterol. So when we expose ourselves to sunlight, what happens? The skin is converting 7 dehydrocholesterol to cholecalciferol. Okay, that is D3. Okay, so in the presence of this calciol 25 hydroxylase enzyme. So cholecalciferol is converted to 25 hydroxycholecalciferol. So this first step is taking place in the liver. So second activation, you see calcidiol, it is getting transported to kidney via blood circulation, and here you will see calcidiol 1 alpha hydroxylase is converting this calcidiol to 125 dihydroxycholecalciferol, that is nothing but calcitriol, active form of vitamin D. So if we expose ourselves to sunlight, obviously our skin itself will synthesize natural active form of vitamin D. Hence vitamin D is called as sunshine vitamin. Okay, so once you write this natural biosynthesis of vitamin D, then we should know the biochemical functions. So when we were doing calcium metabolism, when I told you like calcium is very like how is the calcium regulated, I always used calcitriol is promoting reabsorption of calcium from renal tubules, isn't it? That means calcium is actually blood calcium levels are maintained because of the reabsorption of calcium from nephron in the presence of calcitriol. Calcitriol is nothing but active form of vitamin D, 125 dihydroxycholecalciferol. One more mechanism, same thing is given in different way. Beta, before I go to functions, let me finish this. So in the skin, we have 7-dehydrocholesterol. In the presence of UV light, UV rays of sunlight, it is converted to cholecalciferol. Liver converts it to 25 hydroxycholecalciferol. Kidney converts it into 125 dihydroxycholecalciferol, which actually helps in the reabsorption of calcium from the renal tubules and also from the intestines. Intestinal absorption of calcium is also increased. So calcitriol is actually mobilizing bone calcium, isn't it? Suppose if blood calcium levels are low, you will see the bone calcium will be mobilized and it will be allowed to enter into the plasma. That's what is bone resorption. But suppose if calcium levels are high in the blood, then calcium will be allowed to get deposited in the bone. That's what is bone formation. So that means when calcium levels are low, the bone calcium is mobilized. Okay, so this calcitriol, active form of vitamin D, it is actually binding with the receptors present in the cytoplasm. So it forms like same like hormonal action, you see. Hormones also will have receptors either on the cell membrane or in the cytoplasm or on the nuclear membrane. I told you when I was discussing hormones. So calcitriol is actually binding with the receptors which are present in the cytoplasm. So once calcitriol receptor complex is formed, it is entering into the nucleus and then it is controlling transcription, that is the formation of mRNA. From mRNA, you will see translation taking place. That means formation of proteins. Hence, vitamin D is called as hormone because its function is similar to hormone. Even vitamin A also, I told you the same thing, if you remember. Vitamin A is also considered as hormone because it has receptors in the nucleus. It also helps in the formation of mRNA, then proteins. Isn't it? That means it promotes transcription and translation. Hence, vitamin A and vitamin D, according to new terminology, they are also called as hormones. Okay, now let us see the functions of vitamin D. So basically it is helping with the calcium absorption, so that we have studied in the flow chart, like calcitriol absorption of calcium from the intestines, like action of calcitriol on bone. If calcium is more, it helps in bone formation. If calcium is less in blood, it also causes bone resorption and mobilizes calcium from the bone into the blood. Okay, it helps in the reabsorption of calcium from the renal tubules of nephron. Okay, so that is all written in the theory way. So, so vitamin D is a hormone and not a vitamin. Justification, because it has receptors in the cytoplasm. It helps in transcription, translation. That information will be given in this paragraph. So how much like vitamin D we should take per day? 400 international units or 10 milligrams we can take better. Okay. So or you can see like vitamin D is 200 international units. Okay. So what are the sources of vitamin D? Exposure of skin to sunlight. Okay. Then consumption of natural foods by irradiating foods. That means if you keep certain foods in sunlight like yeast, they are also good sources. Milk, butter, etc. So they are also actually not so good, but still good sources of vitamin D. So what happens if there is deficiency of vitamin D? So it will lead to a condition. Suppose if epiphyseal cartilages are still existing, okay, in a very young bone, then you will see bony deformities. You call it as rickets. Okay, bow legs, knock knees, chest also will become pigeon like. Okay, and coastal cartilages also will become thick, that is rickety rosary. Okay, so all that clinical conditions you see, and the bones will become very soft. You call that as osteomalacia. In case of adults, we see osteomalacia because epiphyseal cartilages won't be there, and because vitamin D is not there, calcium deposition in the bone will not happen, so that the bones will become very much soft and they are actually prone for fracture. So that condition is called as osteomalacia. In case of small children, you see bow legs, knock knees, pigeon chest, rickety rosary. That type of conditions you see in case of small children. Whereas in adults, the bones will become soft. You call it as osteomalacia. Okay. So that is all about vitamin D, beta. So they have given it in a very indirect form. So what is the diagnosis? So probable diagnosis, because she is 50-year-old. So what is the diagnosis you have to give? It is osteomalacia, isn't it? So synthesis, we have to write biochemical functions, we have discussed sources, we have discussed RDA also we have discussed, and lab assessment, that means how to assess, that means blood levels we have to check better, but blood levels checking is not given here. You have to check in the internet. So what is the test which is usually done? Normal levels also we should write better. Blood vitamin D levels. Okay. So have you gone through other questions? So in case of glycosaminoglycans, so they are nothing but heteropolysaccharides. Polysaccharide means what? So if we have more monosaccharide units, generally we call them as polysaccharides. Like starch is a polysaccharide, glycogen is a polysaccharide, isn't it? Because it has many units of glucose molecule. Many units of monosaccharide makes polysaccharide. So if polysaccharides are in combination with either lipid or with protein, we generally call them as heteropolysaccharides. Okay. So under heteropolysaccharides, we have mucopolysaccharides, which are also called as glycosaminoglycans. That means polysaccharide will be in combination with some amino sugar and uronic acid. So it's better we take the help of pictures to make you understand. So for glycosaminoglycan, I want every one of you to learn this table. Okay, we have hyaluronic acid, chondroitin sulfate, heparan, dermatan sulfate, and keratin sulfate. You need not learn the pictures, but to make you understand, generally you see this is the structure of hyaluronic acid, which is made up of D-glucuronic acid and N-acetyl glucosamine, and it has beta 1,4 glycosidic linkage. So suppose if straight linkage, generally you call it as alpha 1,4, but if it is cross, then it is beta glycosidic linkages. So chondroitin sulfate is made up of D-glucuronic acid and N-acetyl glucosamine, and so on. everything composition is given structure wise. Now, for example, hyaluronic acid composition, we have already seen what is it made up of? D-glucuronic acid and N-acetyl glucosamine, same thing. So where do we see hyaluronic acid? We see in connective tissue, synovial fluid, and vitreous humor of eyeball. So functions, it serves as a lubricant and shock absorber, and it promotes wound healing. Okay, so this is the thing you have to write under hyaluronic acid. Similarly, we have many other like chondroitin, heparin, dermaton sulfate, and keratan sulfate. So this is very, very important question, better. Glycosaminoglycan. Okay, so nucleosome we will go with next question.